Hi, my name is Abby Knight, and I'm one of the 2016 Arnold O. Beckman Postdoctoral Fellows. I've been pursuing my postdoctoral research here at the University of California at Santa Barbara in the laboratory of Professor Craig Hawker. My postdoctoral research is focused on my interest in biomimetic materials, in addition to the strengths of my research group and my institution in polymer chemistry and materials characterization.
I want to start by describing Arnold O. Beckman's contributions to science, including the development of tools such as the pH meter, the spectrophotometer, and this oxygen analyzer. Each of these characterize chemical properties of the environment, including things like acidity and basicity and the transmission of light. Biological nanostructures, such as proteins, respond to environmental stimuli, including those that Dr. Beckman was studying. What I've shown here is a linear structure of a protein composed of amino acid building blocks. I've highlighted the interactions between them that lead to the three-dimensional structure, such as this barrel structure shown here. Slight changes in the environment can alter these interactions, leading to structural changes. This can be important for functions such as cell signaling, transport of small molecules, and even catalysis. The dynamic nature of these proteins can be required for even the most routine things in daily life.
Synthetic chemists and material scientists have strived for decades to mimic the functionality of natural proteins. However, the toolbox of stimuli for making stimuli-responsive materials is currently limited. To add to this toolbox, I have synthesized materials that respond to the presence of metal ions. These materials are inspired by molecules secreted by marine bacteria, making this location particularly appropriate.
Many bacteria secrete molecules called siderophores in order to recruit iron that they need for their metabolism. Interestingly, marine bacteria secrete siderophores that are amphiphilic. They have a hydrophobic or water-insoluble and hydrophilic or water-soluble portion. This allows for self-assembly into the spherical micelles shown here. What's particularly interesting about these marine siderophore amphiphiles is that they are able to undergo a morphological shift in the presence of a bridging metal ion, forming this hollow spherical vesicle. This morphological shift is something that we wanted to be able to mimic synthetically. So we designed a synthetic amphiphile that has a hydrophobic portion made up of a small fragment of polystyrene, which is something you might be familiar with from a styrofoam cup. And then for the hydrophilic position, we have a peptide or a fragment of a protein. The first thing we wanted to do was to visualize the assembly in the absence of any metal ions. This is an image taken with cryogenic transmission electron microscopy at the molecular foundry of our amphiphile assembled in water. Here you can see we formed vesicles, so again spherical structures, as indicated by the white arrows. We were excited to see that upon the addition of divalent ions, including cobalt and zinc, we were able to form a different morphology. So instead of forming isolated spherical structures, we formed smaller particles that are aggregated together in a chain-like structure, again indicated by the white arrows. In the presence of divalent ions, including nickel and cadmium, we were able to visualize isolated spherical micelles. The final nanomaterial we were able to visualize was formed in the presence of divalent manganese. Here, the structures again are isolated spheres, but these are actually multilamellar vesicles, meaning they have multiple layers, kind of like an onion. In this work, we're excited to demonstrate that we can synthesize a synthetic amphiphile that forms different nanostructures that are only directed by the presence of different divalent metal ions.
I would like to acknowledge contributions from scientists at the Molecular Foundry at the Lawrence Berkeley National Laboratory and my colleagues within the Hawker Group. I would also like to sincerely thank the Arnold and Mabel Beckman Foundation for supporting this interdisciplinary research. I intend to continue these research directions in my independent career as a professor in the chemistry department at the University of North Carolina, Chapel Hill, where I first joined the Beckman family as a Beckman Scholar as an undergraduate student. It's been incredibly rewarding to return to the annual symposium and engage with the students, postdocs, and faculty. Thank you again for your continued support.